I'm going to talk about the preclinical dengue vaccine development, and I'm going to talk about this in broad terms, covering starting at the selection of parent seed viruses, passage and culture of those viruses, freedom from adventitious agents, predictors of safety, formulation and dosing strategies, and if I have time, I'm going to cover risks to the environment as well. And my talk's going to be in two parts. First of all, I want to describe the preclinical challenges of dengue vaccine development from the perspective of the virus and host. And then in the second part, I'm going to speak about Sanofi's preclinical experience with the two candidates that they've worked on so far. Now, the goal of vaccine development, of course, is to make a safe and effective vaccine, and some of the challenges are shown on this slide. Many of you have seen this slide before. So the absence of either an animal model for disease or a recognized immune correlator protection creates challenges for the selection of suitable vaccine candidates, and also for the standard measurement of vaccine potency. And as a result, we become, on the one hand, more reliant on the outcomes of clinical trials, but also, on the other hand, on the physical, immunological, and biochemical characterization of the vaccine to identify key features that, when combined, allow us to reliably predict the safety and efficacy of the vaccine. Now, these key features need to be expressed within stringent limits, and these limits are defined from the variation of batches that were proven to be successful in clinical trials. So, and then a further challenge, of course, is to be able to reproducibly manufacture vaccine that meets these stringent limits. Sorry, these are animations that I want to move. Okay. So to begin to describe the key features that define safety and efficacy of the vaccine, we need to understand some specifics of dengue viruses and how they cause infection, and then how the virus interacts with the host to cause disease and immunity. So beginning here with the virus, dengue virus is a member of the flaviviridae family. This is a membership that it shares with other human pathogens for which we already have live attenuated vaccines, such as yellow fever and Japanese encephalitis. And that gives us some hope that we will be able to develop a vaccine for dengue. Now, as we all know, there are 4 closely related but antigenically distinct serotypes of dengue, dengue 1 through to 4, and different genotypes of these viruses have been described, and there appear to be differences in virulence and epidemic potential related to differences in these genotypes. Nevertheless, infection with one serotype leads to long-lasting protection with that same serotype. Now, it's an envelope virus. It has a positive sense RNA genome. The genome codes for 3 structural and 7 non-structural proteins. The virus enters the cell by receptor-mediated endocytosis. Now, either directly through a binding site thought to be on domain 3 of the envelope protein, or through antibody bound to virus. Virus replication occurs in the cytoplasm of the cell, and maturation of the virus occurs at a late stage through cleavage of the pre-membrane protein. Now, this is all important. Maturation of the virus is incomplete, and when you look at the electron microscopy of virus preparations, you can see that they're a mixture of mature and immature particles. Mature particles can infect cells directly through the binding domain on the envelope protein, but both mature and immature virus can infect Fc gamma receptor-bearing cells in the presence of antibody. It's also important to note that viral proteins need to be correctly glycosylated, and nitrogen-linked glycosylation affects the structure and function of these proteins and has been shown to affect infection, genome replication, and progeny virus packing of dengue viruses. So correct glycosylation of the envelope protein impacts the ability of the virus to infect cells and replicate. So both the electron microscopy appearance and the protein glycosylation as determined by gel electrophoresis are two key features that need to be characterized for the vaccine virus. The virus genome is duplicated by RNA replicase, and this lacks the proofreading ability of DNA polymerase. So replication of the genome is an error-prone process. Mutations are unavoidable across multiple passages of the virus, especially for live attenuated strains obtained empirically on different cell substrates. And mutations that occur can be associated with either an increase or decrease in virulence of the virus. So it's important to fully characterize the genetic sequence of the vaccine virus at all stages of virus propagation and production. Okay. So it's also important to understand how the host is likely to react to the vaccine. And to do this, we need to understand as well as we can, and it is quite difficult, how the host responds to and develops immunity to the wild type dengue virus. Now, the major phagocytic cells of the innate immune response, the monocytes, macrophages, and dendritic cells, are both permissive to dengue virus replication and responsible for detecting and removing virus. They also initiate the adaptive immune response through antigen presentation to T lymphocytes, and T lymphocytes in turn secrete cytokines that activate macrophages and cause them to kill virus. Dendritic cells are the most permissive cells for dengue virus replication, and the dendritic cells in the skin are the first that virus replicates. Now, primary infection, particularly in children, as we've seen, usually results in uncomplicated dengue fever or asymptomatic infection, and the development of both humoral and cellular immunity that protects against disease with that same serotype. Now, but the anti-dengue immune response, as we've also seen discussed at this meeting, is by nature cross-reactive. But cross-protection against other serotypes is not long-lasting. An estimate in a modelling paper that was published this year suggests that this can be anything between 1 and 3 years, this cross-protection. And in fact, of course, secondary infection is associated with a greater risk of severe dengue. Now, the CD4 Th1 type response is important for protection against viruses, and this is characterized by production of antiviral interferons. Cytotoxic T cells are also important because they destroy virus-infected cells. There's also a phenomenon known as original antigenic sin, and again this has been mentioned at this conference. And this affects the in vitro observed humoral and cellular immune responses to dengue viruses. So it's clear that severe dengue is an immune pathology involving both cellular and humoral arms of the immune response, but the precise conditions leading to severe disease in the individual are still under investigation and clearly complex. So in terms of vaccine development, it is important to characterize the cellular and humoral immune responses to vaccine. And for us, this has meant ensuring that the vaccine virus has an immune response profile that is likely to stimulate virus clearance without provoking pathology. So our emphasis has been on generating a vaccine that will be safe for administration. And so now I'm going to talk about our experience with 2 vaccine candidates. The first is the one that was developed by Mahidol, and then the second one is the vaccine that was developed by Acambis. And so this is the first vaccine candidate that we explored. The vaccine viruses were provided at the pre-master seed lot by Mahidol University. where they had been attenuated by serial passage in cell culture. Dengue viruses 1, 2, and 4 had been passaged in primary canine kidney cells, whereas dengue 3 was passaged first in primary green monkey kidney cells, then in primary fetal rhesus lung cells. Now, trials with this vaccine in Mahidol University in Thai children showed some promise, but there was an imbalance in serotype 3 response. The partnership was set up with Sanofi, and we adapted these viruses for growth in vero cells, but we nevertheless abandoned this candidate due to under-attenuation of the serotype 3 and stability issues with serotype 4. Now, what this slide demonstrates quite nicely, I think, is the variety of different cells, substrates that can be used for early parent seed lot development. And I want to just emphasize that it is essential to meticulously record the passage history of viruses used for vaccine development, including the cell substrates used, the animal products used in culture media, and indeed the types of laboratories in which the viruses were isolated and grown. And if you look at the FDA guidance on these matters, all of this is very well stated there. Good laboratory accreditation of laboratories is also important. So now I'll just talk about the Chimerivax technology that was developed by Cambis and the work that we've done with these vaccine viruses. So this slide summarizes our overall characterization of the vaccine including the work done by Cambis some 14 years ago. So this has been an extensive work. It's important to remember that the characterization tests that are done are both specific for the general risks and challenges posed by dengue viruses and also the specific risks for the particular virus construct. And so today, I don't have time. Obviously, this is extensive work to go into all of the data that's been generated. I'm going to just emphasize 5 aspects of this development. I'm going to talk about the genetic and phenotypic stability. I'm going to talk about freedom from adventitious agents, tissue tropism of flaviviruses, immune response assessment to predict safety, and environmental risk assessment, if I have time, because this candidate, of course, is a genetic construct. Okay. So this slide shows the construction of these chimeric viruses. So 4 genetic constructs were created, 1 for each serotype. They're all based on the same yellow fever 17D backbone with insertion of the pre-membrane and envelope genes from each of the dengue serotypes. Now, the parent viruses for these constructs were actually isolated some 30 years ago. Serotype 1 and 2 was isolated in 1980, serotype 3 in 1988, and serotype 4 in 1978. And each of these viruses was originally isolated in mosquitoes or mosquito-derived cells, passaged a couple of times before amplification and cloning and sequencing of the pre-membrane and envelope genes, and then construction of the chimeric viruses. And all chimeric viruses were plaque purified to select strains that would be used for further vaccine development. Now, if I might just step to the side a little bit here, and this is not what I have a slide to cover for this talk, but just to mention that, of course, we have conducted some phylogenetic analysis of the strains that were circulating in Ratchbury during the phase 2B study that we've recently concluded, and compared that analysis with the genetic analysis, the phenotypic analysis of the virus used in the vaccines, and we can say that there is no association between genetic distance and the efficacy observed in the study. And for example, the circulating serotype 4 virus in Ratchaburi is a genotype 1 virus, whereas the vaccine virus is a genotype 2 virus. For serotype 2, both the vaccine and circulating genotypes are Asian type 1. Now, as I've mentioned, flavivirus has a positive sense RNA genome. This is duplicated by RNA replicase, which is error-prone. Mutations are unavoidable, and so it's important to determine the genotypic and phenotypic stability. Nevertheless, the seed lot system that was used for yellow fever vaccine has been remarkably successful in maintaining the attenuated phenotype of the yellow fever 17D vaccine over the decades, and this high genetic stability may be attributed to the low error rate of yellow fever 17D virus RNA polymerase. It is important to have a very well characterized seed lot system for the viruses and to have clearly recorded the transfection and cloning steps, including the use of all cell cultures and culture media used, as I've already said. And this is really important because this is what the regulators will look at when they assess how well you've validated freedom from the presence of adventitious agents in the vaccine. So, and in terms of genetic stability, we've studied the full genome sequence for all 4 viruses from the very first passage and at the pre-master seed level, the master seed level, and at the bulk stage, which is used for vaccine manufacture, and at 10 passages beyond bulk. And we see high genomic stability with only 9 point mutations, and most of these at a stage beyond a bulk manufacture. In addition, we've looked at phenotype of the viruses. We've looked at plaque size phenotype. This is stable at all production steps. And the virus morphology observed by electron microscopy is typical for flaviviruses at each stage of manufacture. There is also a consistent ratio between non-infectious and infectious particles, and this is something else that the regulators will look for. And in fact, it's consistent with what we've seen with yellow fever vaccine lots. Now, as well as characterizing the viruses, it's equally important to characterize the cells that you use to grow the viruses in. And this is important, of course, for safety and for demonstration of freedom from adventitious agents. You need to be able to document where the cells come from and how they have been maintained prior to use for the manufacturing process. And you need a well-defined seed lot system for these cells. And we use Vero cells. There are a number of advantages for these cells. They are very well characterized. They're widely accepted by regulatory authorities. They grow really nicely in bioreactors, and you can get high yields of vaccine virus. And they've been used successfully over 30 years with the polio and rabies vaccines. Now, the preclinical stage is a stage where you try you demonstrate the safety of the vaccine. It's also a time where you can begin to assess formulation and dosing strategies. And in particular for dengue viruses, of course, viral interference is a problem for tetravalent vaccines. and this can result in an imbalanced immune response. So in the early development of this candidate, the team at Acambis used macaques to test preliminary vaccine formulations for viral interference in terms of immunogenicity and viremia. Now, although they did see differences between the different virus serotypes in terms of viremia and antibody response in these experiments, and there was dominance of serotype 4, the results demonstrated that a balanced immune response could be achieved with schedule adjustment and boosting. Now, dengue is not considered generally to be a neurotropic virus. Nevertheless, flaviviruses are capable of causing a variety of diseases with different organ specificity. And because of theoretical concerns, it is necessary to study carefully the tissue tropism of the constructed viruses using in vitro and in vivo tests. And for example, we've tested the growth characteristics of different human cell lines of the virus in different human cell lines, and in fact the virus grows to significantly lower titers than the yellow fever 17D vaccine virus in human hepatic cell lines. And in tests in macaques and in our mouse neurovirulence model, we've demonstrated that the vaccine viruses are safe and significantly more attenuated than the yellow fever 17D virus, which of course is universally accepted for its safe and effective use in humans. Now, this slide summarizes the work that's been done over the 14 years to characterize the immune response. Our concern has been primarily safety, as I've said. It's been to establish that vaccine viruses would produce an immune response that was consistent with protection and low reactogenicity when the vaccine was used in humans. Now, to explore the innate immune response, we infected flavivirus-naive human dendritic cells with a vaccine virus in humans. We observed maturation of these cells with upregulation of pro-inflammatory cytokines, including interferon gamma, but with lower levels of tumor necrosis factor alpha than was seen when these same cells were infected with parental viruses. So, in terms of the innate immune response, the vaccine virus induced maturation of naive dendritic cells and a controlled response accompanied by limited inflammatory cytokine production and consistent expression of antiviral interferons. Now, in terms of the adaptive immune response, in vitro stimulation of vaccinee cells produced a dengue-specific T helper and CD8 response, both characterized by interferon gamma to tumor necrosis factor alpha ratio in favor of interferon gamma. So there was no Th2 response observed, and the serotype specific response in these experiments was again dominated by dengue virus 4 after the first vaccination, but a broader response was seen after boosting and in dengue virus 1 and 2 vaccine primed individuals. And of course, we've gone on from preclinical work here to look at the immune response in vaccinees, and in human trials with the tetravalent vaccine, there have been no detectable changes in serum pro-inflammatory cytokines, whatever the vaccinee's prior immune status. And this is consistent with the low level of observed reactogenicity between to the vaccine. It's notable that the CD8, so the cytotoxic T cell response, is directed at the NS3 protein, and there's no cross-reactivity between dengue and yellow fever NS3. Now, this is a vaccine that's produced by recombinant technology, and so it's necessary to conduct an environmental risk assessment in compliance with national guidelines. And in fact, we've proven the environmental safety, the concept of the environmental safety with this construct, because our Japanese encephalitis vaccine has the same construct and has been already approved for use in Australia and Thailand. So it's important to note that we've established a number of things, including that the level of viremia following vaccination is less than the infectious dose for mosquitoes, so it's unlikely that mosquitoes will become infected with vaccine virus. And even if they did, the recombinant viruses do not replicate in mosquitoes, even when they're fed to the mosquitoes at high doses. And that in the worst case scenario, artificially constructed recombinant wild-type yellow fever and dengue virus chimeras are substantially attenuated relative to vaccine or wild-type strains. And so we've been able to conclude negligible environmental impact from this chimeric vaccine. And this is my final slide, and it summarizes some of the characterization work that we've done for the development of our current vaccine candidate. My key message today has been that vaccine candidates have to be very well characterized during the preclinical stage of development to address both dengue virus specific and vaccine specific issues that predict the safety and efficacy of the vaccine. These issues include the genetic and phenotypic stability of vaccine virus, a careful description of the physical and biochemical properties of the viruses, absence of adventitious agents, tissue tropism of the viruses, an assessment of the immune response to predict safety, and finally an assessment of the environmental risk. And these characterization steps are particularly important because there are no animal models for disease, and there's currently no established correlate of protection. So I'd like to thank you all for listening to me today.
Thank you, Jeremy Brent. The last speaker will be